Hello, grade 10 students. In this presentation, we're going to talk about synapses and the different type of actions inside our body. To start with, let's start with the types of actions. We have actions that require thinking, others they do not require thinking. Some of them they require the intervention of the brain in addition to the spinal cord, others they don't require the intervention of the brain. These are called voluntary actions that require thinking. Those that doesn't require thinking, they are called involuntary, or they have a name is called reflex. Sympathetic and parasympathetic. Parasympathetic nervous system PNS controls homeostasis and the body at rest, and is also responsible for the body's rest and digest function. Sympathetic nervous system (SNS) controls the body's response to perceived threat and is responsible for the fight or flight response. These two, parasympathetic and sympathetic, they are part of the autonomic nervous system, which is responsible for the involuntary actions inside the body. A neuron or nerve fiber is said to be at rest in the absence of any stimulations. So, aim of the following experiment is to study the electrical state of a neuron at rest. So here we have in experiment 1, we have a nerve fiber where the electrodes are found on the external of the nerve fiber where the delta V was 0 millivolt. The delta V was 0, meaning that the needle of voltmeter didn't deviate. What does this imply? This implies that there is no potential difference existence on the surface of the axon, or there is no potential difference at all on the surface of the axon. In experiment 2, we have the same experiment, but the needles they are placed on the internal side of the axon. Also, the needle doesn't deviate, meaning that there is no potential difference in the internal medium of the axon. Internal meaning that inside the axon. Let's move now to experiment 3. Experiment 3, one of them is placed in the outside, external, and the internal. The delta V, the potential difference, is minus 70. What does this mean? This means that there exists a potential difference. We can say that we can deduce from these experiments the following. In neuron at rest, the potential difference existing between the interior and the exterior of the nerve fiber delta V does not equal to 0. As said, the nerve fiber is said to be polarized. Delta V less than 0, the interior is negatively charged with respect to the exterior. The Raster potential is the potential difference that exists between the intracellular and the extracellular of a nerve fiber at rest. So, note here that resting potential may range between minus 5 to minus 90 depending on the species and the nature of the nerve. To keep in mind, you have some notes, you have to know it. We have some notes. First, excitable neuron, a neuron that's able to respond to an effective stimulus. So action potential is the term given to the electric visualization of the response of a neuron. Let's move to other notes. We have threshold of excitation is the minimum intensity of stimulation that's able to create or to cause an action potential. Below this intensity, there is no response or there is no action potential. Also, we have upon excitation, a change in the potential difference occurs the interior and the exterior of the nerve fiber should occur and should reach the threshold of depolarization in order to initiate an action potential, meaning that initiation of the response. Threshold of depolarization is the minimum change of the potential difference that should be reached so that to create an action potential. Also, we have minimum time of excitation is the minimum time during which an effective stimulation could cause or could initiate a response like in urine. Action potential in nerve fiber is said to be at rest at minus 70 millivolt. This is resting potential upon application of stimulus of intensity I1, which is less than the threshold of excitation. There is no difference, so no change in the action potential as nothing if occur. So the stimulus is said to be ineffective. I2 less than the threshold, but it's greater than I1. Also, there is no, we have a small change, it's artifact. This is less than the threshold, so slight change in the potential difference. It's called artifact. Did not reach the threshold of depolarization since the depolarization is not reached, so there is going to be go normal to the normal state, which is no, and the stimulus is said to be ineffective. Upon application of I3 greater than the threshold, here we have an action potential. Action potential means this stimulus is said to be effective since it reached the action potential. It causes an action potential, and it's the first one to cause an action potential, so it's called the threshold of excitation. Simulation I4 greater than the threshold and it's greater than I3, so we have an action potential. What does this mean? This means that I4 is the effective stimulation since it causes an action potential. All or none, if axon is stimulated above the threshold, it causes response. An axon cannot send a mild or strong response. It either does or does not cause a response. Here we have to know that the nervous message propagate in a unidirectional manner. Dendrite, cell body, axon, then the next cell, dendrite, cell body, axon. Factors that affect the propagation of the nervous message in the nerve fiber, we have drugs. Drugs, stimulants, they accelerate the nervous message, like the coffee, it's a stimulant, while tranquilizers, they decelerate, they decrease the speed, like the sleeping pills. Synapses is the functional connection between the neuron and the following neuron on the other cell, neuron, gland, or a muscle cell. The synapse, they are of different types. We have different types. Before the synapse, we have presynaptic neuron. After it, we have post, and the middle we have the connections called the synaptic cleft. Synapses, according to their structure and according to their function. Classification of synapses, we have neuronal between two neurons, neuromuscular between neuron and muscle, neuroglandular between a neuron and a gland. Neuroeffector synapse, we're going to talk about them in the coming slides. According to the structure, we have neuronal, neuroeffector synapses, neuronal between two neurons, neuroeffector between a neuron and an effector. This is the neuronal synapses, neuronal also. Maybe it's a neuronal, maybe axodendritic between an axon and dendrite, maybe between two axon and between a soma, axosomatic, between, for example, two axon, it's called axonal synapse. Neuroeffector, maybe it's between a neuron and a muscle, it's called neuromuscular endoplate, and it's called motor endoplate. We have neuroeffector synapse, it's between a neuron and a gland. Classification of synapses, neuroeffector synapse So either neuromuscular, as we said, is called motor end plate, and neuroglandular synapse. According to their function, we have excitatory synapse, inhibitory synapse. Excitatory synapse and inhibitory, what do? Excitatory, they allow the propagation of the nervous message from presynaptic to the postsynaptic neuron or postsynaptic cell, while inhibitory, they inhibit the message from the presynaptic to the postsynaptic cell. Propagation of nervous message through a synapse, it occurs by the help of neurotransmitters. Neurotransmitters, they are chemical mediator between two synaptic cells. It's released by the presynaptic into the synaptic cleft where it binds to specific receptors on the postsynaptic cell, especially on their membrane. Here we have the propagation state of conversions where the messages they are converged into one, and we have when the presynaptic neurons they carry nervous message to one synaptic cell, to one postsynaptic cell in specific. State of divergence when one message is going to be diverted into many postsynaptic cells. Thank you.